Try to leave.
So okay? I have one more call. So shall we start now?
Yeah, go ahead.
Okay, good. Yeah. First, I think I have to introduce myself. So my name is Fai Au. I'm from University of Iowa. Actually, if you do not know me, but last week you may know me because actually I present 1 minute slides. Thanks for David's 1 minute. I present my openings, and yes, I show it again. So if you are interested in the position in our labs, let me know. But now we come to the end of our talk, acknowledgement first. Yeah, because I really want to thank a couple people, a couple speakers last week. Mike, David, and Brian, because actually they gave wonderful talks. All I want to talk about today are very related to what they talk about. Leo talked about the transcriptome analysis, how to quantify, estimate the gene isoform abundance. And Mike talk about the advance of long read. David and Brian give wonderful talk about the phasing problem. So because of their talk, I can save a lot of time from giving the detailed introductions, the biological backgrounds. So I really want to thank those, and you will see how we will mix up their talk together and make my talks. At first, we talk about phasing. Brian and David talk about phasings last week. And here an example of phasing problems. Assume you have some sequencing data. You have 2 SNPs here, 2 SNPs of these genes, and then make a guess what's the haplotype of these genes. Problem set. And anybody give it a try? David, you are the expert of phasing. Give it a try.
Try what?
Guess what is the haplotype of this gene?
Oh, you colored it already.
Oh, yeah. Right. I'm so sorry. Yeah, because actually this is part of our publication, so we already colored it. But you will find that, well, let's check. Well, in the SNP1, the major alleles is C because you have 84 read supporting C here and only 42 read supporting T, right? So this is the majors, and the SNP2 G will be the major. So if you make a reasonable guess without this color information, well, CG and TA will be the haplotype, the pair of haplotype you will make a guess. That's a very reasonable guess, right? And if this is the haplotype, and then you can calculate the abundance imbalance between the allele, so-called the allele-specific expression, you can sum up the coverage from the major alleles and then divide it by the total coverage. And the rho representing the allele-specific expression here is 0.65. So is this correct? No. Well, if we check the PepBio data, we have many PepBio data, and 72 very high quality PepBio data, the long reads, covers this region and show that TG and CA is the true haplotypes. So this is not, well, if you do the phasing problems according to the coverage, it's not true. But why? It doesn't make sense, right? Now Leo come out and say, well, remember this is a transcriptome data. So when you take care of the transcriptome works on the transcriptome data, you will have some very complicated scenario like this. Let's talk about what is transcriptome first. Well, although they have given wonderful talk, but I would like to use 1 minute, one slide to introduce transcriptome. You have a gene. This is the DNA sequence of a gene, and after transcription, you have mRNA sequence. But this is pre-mRNA. This is not the final product of mRNA. We have some process called alternative splicing. After alternative splicing, we have a different combination of the exons. So you have exon 1, exon 2, exon 3, exon 4 in these genes. But in the isoform 1, you only express exon 1, 2, 4. And in the second isoform 2, we have exon 1, 3, 4. This is a different combination expression of the exons. So they give you different product of the mRNA. This is the final product. However, after we listen Brian's and David's talk, we know we have 2 copies of genes, maternal copy and paternal copy, because you have 2 copies from your moms and your dad. So now, well, they have only small difference in this position for it's called SNPs. We have AG here, CG here, and TA here. So definitely you have 2 different type of the pre-mRNA, and then you have 2 different type of the isoform. Same here in the isoform, you have two paternal isoform 1s, maternal isoform 1, and paternal isoform 2, and maternal isoform 2. So instead of two isoform product, now you have 4. And now you know what the transcriptome is. Then we come back to these questions. Well, when we only consider this isoform, well, originally in the annotation library, we only have one isoform annotated in the library. However, using the Fai Au data, we find, oh, there's two other isoforms, which is novel. And these two novel isoforms do not express the SNP2, right? We have empty here, this is the intronic regions, and they skip this SNP2. And while the expression level of these three isoforms are completely different, from low to very high, and also they have a different bias between the paternal and maternal alleles. For the first one is 0.6, and the second one is 0.2, and the other one is even smaller. So because this very complicated expression events, so you if you just count the number of coverage in 2 SNPs, the guess will be wrong. So that's why we make a lot of challenges in analyzing the transcriptomes. When we're analyzing the transcriptome, we have to take care of the isoform expression events, and also we have to take care of the haplotyping. So both gene isoform and the haplotypes are pretty important. But when we analyze the transcriptome, we consider these two events, and also we need to do two things for both. We need to identify them. That means we need to identify which isoform expressed, including the novel one, and we also need to determine the haplotype. What is the combination of these SNPs, the alleles, in these genes, and then we can quantify them. So we need to give 6 value, the expression of each isoform and the imbalance between them in the 2 alleles. And actually, it's pretty difficult to identify the reconstruct the gene isoform if we only consider Leo's problem gene isoform. Well, there's a couple of papers show, well, this problem is NP hard if you just use the short read data, Illumina data, and use the maximum likelihood method. This is too difficult to solve. And why we have this problem? Because the isoform is too long. This is the gene isoform length distribution. We have a median length about 2,500 base pair. And what you have on hand is the Illumina data, 50 to 300 base pair. It's pretty short. And then when you want to reconstruct something 10 times or even 100 times longer than that, so you will make a lot of problems. For example, the combination problem. You have to check all the possible combination of the exon. So this is not good. You remember I thanked 4 speakers, and the last speaker come out, Mike. Mike introduced long-way, right? Long-way is pretty useful. Long-way can help us to link the exon, can help us to link the SNP so that we can identify isoform, we can determine the haplotype very easily. So long-way is about few thousand base pair and up to about 30,000 base pair. It's almost cover the full length of the transcript. And the major platform is Fai Au sequencing or Oxford Nanobot technology sequencing. However, well, it gives us very long ways, but the error rate is pretty high. Comparing to just 1% error rate given by Illumina data, error rates of the Fai Au data is about 15%. So it's pretty high error rate. So we have to introduce another term, hybrid sequencing. We use the accuracy, high accuracy, and high throughput of the Illumina data, the second generation sequencing, and also the length advantage of the third generation sequencing together. We call it hybrid seq to solve the transcriptome problems. So in order to solve it, we develop a pipeline to do that. Start with the long way and short way. We develop this pipeline. So we have 7 tools now, and well, if we go through everything, we may need about 2 hours. Very fortunately, I just find that well, we reserved this conference until 4:30, so we still have enough time. Okay, we are going to check one by one. Okay.
It's already 3:40.
Oh really? No, serious. This is the hour time. I'm sorry for that. Well, instead of going through everything, I only pick up 3. LSC for error correction, because we know the error is too high, so we want to use the short way to correct the long way so that you can remove most of the errors in your long way so that you can make use of the long way. And then we have to identify Leo's problems, the isoform problems. Okay, we develop IDP, isoform detection and prediction, and then we apply to the allele-specific expression, David and Brian's problems, how much the paternal alleles and maternal allele expressed in the gene level and the isoform level. So we only talk about these 3. Okay, the first 2, error correction. 15% error is too high to be used. Well, when we make data like this, we have very high quality short V and low quality data long V. It's something like, well, you have a puzzle, right? When you open the boxes, you have very small P6 things, but the resolution is pretty high, but on the top of the box, you have pictures. This picture tells you what you're going to make, but actually, probably you want to make a picture like this big with this small P6. This is exactly the goal what we want to do. We want to have a high resolution long V, right? A high resolution of these big pictures. So when you have a puzzle, what you're going to do first? The second, yeah, this is a good strategy. I'll use it as well. What else? What else? Okay, well, probably we can start with this strategy. Let's pick up this blue one. We know this blue one definitely not come from the bottom left, right? Because there's nothing blue here. We put in the upper corner here. So we know this may come from the lake or come from the sky. So this is very similar to a process of sequence analysis. What's that? Alignment. You want to align something back to a reference, right? Okay. Well, we want to align the high quality short V back to the long V, but we have a basic assumption of alignment, the similarity of the sequence. You have the reference sequence, you have target sequence, they should be similar enough. But when we align this high quality short V back to the low quality long V, we know they are pretty different. Because you have a lot of error here, you do not have the error here. So the alignment process would be a very challenging problem. Well, now you make chicken or egg problems. You want to correct the error, but the high error prevent you from doing the first step. So we give up? No. Well, let's check how the error look like first. In order to do that, we have a tool called Align QC. Well, I'm not going to introduce this tool, but this tool is to do the QC of the long V. One of the output of Align QC is the error pattern of your data, how it looks like in different contexts. This is the upstream one basics, and this is the downstream from that insertion, deletions, or the mismatch. For example, let's take this T. Well, if we have the insertion of T's in somewhere, we'll find that, well, if the upstreams or downstream are T, would prefer to introduce a T here. Well, in particular, in this very high probability spot here, that means, well, if you have TT, upstream or downstream, then you will introduce insert a T here. That means you have this L shape, or this is cross shape, is a homopolymer patterns of the error. The similar thing you can find in the deletion and insert in the mismatch. Well, that means, well, if the original sequence is 6T here, well, it will likely to be sequenced as 4T or 7T. But if we can compress them into a single T, compress the homopolymer, we do not have any mismatch between this now. We only have a single T, right? You compress the error. Now, you compress the homopolymers in the long V's and also the short V. You compress them. Then you can remove some error first in the alignment step so that you can align the short V back to the long V much easier with a higher sensitivity and a fast speed. Okay, now we can do that. And the next thing would be very simple. Here's the flow charts of the LSC. We do the homopolymer compression for long V and short V, and then we align the short V to the long V using bow tie or BMW, the short V aligner. And then you just generate the consensus of the short V to replace the mismatch between the short V and long V. That's it. Very simple. The most challenging part would be in the alignment step. Well, after error correction, after replacing the long V sequence by the short V consensus, we can improve the error much better. You see, this is the accuracy after error correction. We have most of them about higher than 90% accuracy or 95%. Before that, you have a peaks about 85%. Okay, that's good. And also, we show that the more short V you align to a position, the better error correction you will have. You have a short V aligned to a position, then after error correction, the accuracy can as high as about 99%. Right? If we do the homopolymer compression, we can have more short V, aligned to the long. We can help you to improve the error correction. Because if you do not do the homopolymer compression, well, most of them may be filtered out according to the editing distance filter.
So so you do the compression to only 1T, regardless of how many there were, with
Yeah,
no
yeah.
bounds. They
Well,
could have been 250, it's still 1T.
you are totally right. And actually, we try different strategies. For example, we have tried saying, well, if you have only 3 nucleotide, the homopolymer length is 3, and then we compress to 1, and longer than 3, we compress to 2, something like that. It cannot help much better than that.
Okay.
Yeah.
So when you do the alignment, do you relax your insertion penalty? Because it might have really been a TT there.
You're right, but actually we do not change any parameters in the aligners. We just use a default setting.
So even if the accuracy goes up, it might be biased toward...
Yeah, it could be.
Okay.
But overall, I think it looks fine. Well, this is the tool we developed about 5 years ago, honestly. And the computational complexity is n times n. M is the number of long read, n is the number of short read, because you need to do the pairwise alignment. But actually, if we use the divergent graph, we can reduce the computational complexity to n plus n. So it's much, much faster. That will be the next generation error correction. I'm not going to talk about this today. There's too many we can talk about today, but we only pick up a few. Well, and here's an example of the long read before and after error correction. We have some so-called raw long read here. Before error corrections, well, once we align to the reference genome, we have a lot of the orange colors or the red color that the insertion, deletion, or mismatch. You have a lot. But then after error correction, you reduce a lot of this error. More importantly, I highlight here there's an exon and here's an exon. Before error correction, the error rates in this region are too high to be mappable. So if you just map the raw data directly, you will miss these 2 exons. But after error correction, we recover this exon and this exon. You can detect the full-length isoform directly. That's pretty good. Now we have overcome the high error rate problems in long read. We really want to solve Leo's problems. Well, the gene isoform identification. Okay, we want to solve this problem in this dimension. Well, how to do that? Well, we know isoform is a combination of exon. In these genes, we have 5 exons, 1, 2, 3, 4, 5, and we have different combination, make different isoform. And the first one is the isoform 1, 2, the isoform 1 equivalent to the exon 1, 2, 4, 5, and the second one is the all isoform expressed together, and the isoform 3 is 2, 3, 5. So a different combination. So if we know the combination of exon, we know the isoform. However, the isoform identification is not very accurate based on the short read data. And if we use the annotation library of the exon, you'll miss a lot of novel exon, novel genes, novel isoform of the known gene. So this is not good. Instead, we find, well, in the same problem, we can reconstruct the isoform by the junction combination. For the same example, assume you know the 5' n and the 3' n, the street 2 boundary. And what you need to do is to find the combination of the junction, right? The isoform is almost equivalent to 1, 2, 3, something like that. And the good news is, well, based on the Illumina data, well, it's very easy to detect most or almost all possible junction. And it will allow us to find the novel gene or novel isoform of the existing gene containing the novel junction as well. So we will use the junction as the structural units of the gene isoform identification. But how can we find the combination? Well, now long read come out. Well, when we align a long read as what we show in the previous example, you find, well, some fragment align to here, some other fragment align to here. And then here's a gap between them. This is the junction. Now, well, if this junction matched to some annotated isoforms in the library, saying this is the known isoform in the RAPSIC library, then we know, well, this exon, this isoform is detected by this long read. So this very straightforward idea, this is called detection because you can observe it directly. Well, using these detection ideas, we find for the very short genes, we can detect about 80% of the gene isoform. And for the very long genes, we can only detect about 30%. Yeah, it makes sense because well, although the long read is pretty long, but it still cannot detect all the full-length transcript. Some can only detect the fragments of the very long gene. So that's why the detection may go down. So that's why we have to take the second step, prediction. We have to predict the very long, the full length of the isoform. But if you only can detect a fragment of these long genes, How we can do that? We have 3 steps. At first, we reconstruct all the possible combination, the candidates. And next, we estimate the isoform candidate abundance, and then we will select the isoform candidates by the relative abundance. At first, we have to reconstruct the possible isoform candidates. Here's the non-redundant long-way alignment you have. Well, and then you find in this gene we have 7 junctions, okay? And we know the 5' N, 3' N, 2 boundaries of these genes by some other data. Okay, now you're ready to reconstruct that. Because the junction 3, 4, 5, 6 are linked together by this long way, we know you definitely do not have any other possible combination of 3, 4, 5, 6. Because in a high throughput long way sequencing, you only observe this, then you fix them, fix the combination. What remains is the junction 1, junction 2, and junction 7, they are free. That's it. You have to check the possible combination of 1, 2, 7. You have 3 free junctions. The possible combination would be 8, right? You just check the combination and then fill it up. This is the candidates. Well, I remind you that if we do not have long way, how many combinations we will have? 2 to 7, 128. So it would be much easier to pick up the true one from 8 candidates than 128 candidates. Well, it's true. So, but how we going to select it? We'll calculate the estimate the abundance of each isoform. Well, I think Leo gave a very good introduction to some Selenium models, but here I have one slide to introduce that. Well, assumes this is the parameter theta 1, theta 2, theta 3 for the isoform, the abundance. And this is what you want to estimate the abundance. And what you observe is the length of each region. You need to normalize it. And also the number of V mapped to these regions.
So I have a question here. You only have a limited number of long reads, right? So, I mean, how do you know that they sample the space of isoforms?
Well, that's very good questions. And actually in the aligned QC, we have some analysis to see, well, whether you reach about 90% or 100% of the sensitivity. Well, and actually the cost of long way is not very expensive now. It's not very high, so you can generate many data. And also you can use this valid protocol called site selection. You can spend more sequencing power in the very long one so that you can save your budget. So now in our data, if you just spend about $2,000, you can cover higher than 90% of the sensitivity. So that's, I think it's okay now. It's not very low throughput now. Well, now I think in the previous talk, we talk about the percentage models or the negative binomial model. Well, you can replace this Poisson model by negative binomial model as well. But well, we here just use the Poisson model. For the number of V from each region of the exon, we assume it follow the Poisson distribution with the lambda x, and this lambda s is the linear combination of this parameter theta i's, okay, theta h, I'm sorry. And just a linear combination, and then if you take log, and then you'll find this is a convex optimization problem. You can solve it very easily, okay? So now you solve it, assume that you already estimate the theta 1, theta 2, theta 3. Within a genes, we calculate a term called relative abundance of each isoform, I. Well, and each one is, this is the fraction of each isoform contribute to the whole gene expression. Because the isoform is constructed by long V and short V, you remember how we construct the isoform candidates, and the coverage, this xi comes from the short V coverage. This is short V coverage. So if we have a high values estimated from the model, that means they have a better match between the short V coverage and the long V structures. So we use this as the classifiers in our ROC curve analysis to pick up the one for the selected FDR. So, well, we compare to our result to a very famous tool, Leo's tools coupling, use short V only, and we find that when we control the FDR to about 5% or 10%, we have much better sensitivity. Okay, so I think it's very important to control the FDR into 5 or 10% because, well, the valid people have to spend days or weeks or months to validate your results. So we compare the performance here rather than here. Okay, and we.
What's the gold standard here?
Oh, good question. Actually, we have another set of the PepBio data, and we only use the one that can be sequenced by the full length. Yeah, I know this is not a perfect validation because we use the same technology. But so far, there's no gold standards of the isoform identification. This is the only way we can do. But of course, this is an unfair comparison because we use two set of data for IDP, but for coupling, we only use short reads. Right, here is we apply to a very famous biological sample called H1 cell line from the human embryonic stem cell. This is pretty important biological cell lines in the biomedical research. We find a lot of isoform and a lot of novel genes. Well, novel genes, this is pretty important, very significant findings in our papers. Let's pick up the top 10 novel genes. Well, the X value is just the index of our novel genes, and the Y value is their expressions of these genes in the other 16 human tissues, normalized by the expression in the H1 cell line. So that's why the expression of these genes in the H1, the red color, is always 1 because you normalize by it. And then we find most of the novel genes have very, very low expression, almost zero expressions in the other ones, but only have the high expression in the H1. And when we take this number A novel gene as an example, we find quite a few long ways supporting these genes. But when you open the RefSeq annotation library, ensemble annotation library, gene code UCLA, the very famous annotation library, you almost find nothing. But the long way and our prediction show that, well, here's a gene with a very complicated exon combination. Okay? And our collaborator did some follow-up, knockdown experiment, and we find 3 of these novel genes. In total, we named 24 novel genes HPAT, the whole human polypotent associated transcript. 3 of them have very strong interaction with the famous the polypotency markers, the profile F1 analog and SOC2. They have strong interaction in the networks. And well, if we knock down one of them, then the polypotency will go out. And you may ask a question that why you can find these important novel genes but not the others technique or not the other papers, because this cell line has been studied for more than 10 years. Because there's a lot of repetitive element in the novel genes. The repetitive element fraction is about 70% or even as high as about 95%. That means most of the regions in the genes is repetitive. Repeat. So saying this is one of the example. Well, if you have a short read align generated from this region, when you align it back to the genome, it's multivariably mappable. So it will filter out. So in the typical RNA-seq data analysis, you just ignore it. Well, even you increase the sequencing depth, you're just increasing this charge in the analysis. So this cannot help even you increase the short read sequencing depth. But once you have a long read covering the repetitive elements, but also some part uniquely mappable, the whole thing is uniquely mappable. Okay? I think Mike presented a similar idea in his talk. But well, you can see the powers of long read now. This long read can help you to confirm the expression of some novel genes, although the novel gene got a lot of repetitive elements. That's good. And this paper has been published in Nature Genetics and highlighted by Nature as well, showing that how we use a new technology and new method to find the dark matters in the transcriptome. Okay? And now we have solved that dimensions, and we have to take care of the other dimension, the horizontal dimension. We have to identify the haplotype, determine the haplotypes, and quantify the allele-specific expression. So what is allele-specific expression? Again, I spent half of this slide to recall what is the allele-specific expression. You have 2 copies of genes in a genome, so paternal and maternal copy, and they only have a small difference in the SNP position. And in the transcription, they have the imbalance of, they do not express as the same. For example, the paternal copy expresses 4 molecules, and maternal copy expresses 2 molecules. So take this toy example, we can calculate the role, the allele-specific expression at the gene level, 0.67 and 0.33. Okay, so this is what you want to estimate. Of course, before doing that, well, we assume you know the haplotype, but in most cases, you don't know the haplotype. So you have to determine the haplotype first. This is the identification problems. Well, now the typical idea is to use short read to do that. Well, you have the short read, the high accuracy short read can determine the genome types. What is the pairs of the SNPs here? What's the pairs here, 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 but you don't know it's the combination of them. Because, well, a short read is too short. It's just about 100 base pair, 200 base pair, 300 base pair. However, by average, the distance between 2 SNPs is about a few hundred base pairs or up to a few thousand base pair. So that is a single shot, we can only cover a single SNP. So if you you find the genome types here, and then you just make a random guess as what we talked about just now, and this will be the successful way because, well, the possible combination is 2 to m minus 1. So you find this go down very quickly. When you have 2 SNP, you only have half chance because you have 2 possible combination, and then when you have 5 SNP, you have 6% successful rate. That is not good. So we introduced long read. Well, in this example, you have some green long read, and this long read can cover the whole gene bodies, or maybe a significant fragment of the gene body can link the SNP together. So it can help us to determine the haplotype. However, well, you see some nucleotide I highlight in the red color because they are the error. So in this position, we should only have A or Gs, but you have T here because of the high error rate. So that is not good. And the long read is not really good for the quantification because you have some bias in the wet lab experiment. So that is not good. So if we only use long read, we cannot do the quantification, but we want to solve both problems. So again, hybrid 6, we use both long read and short read together. And how we can integrate the hybrid 6? Well, at first, we call the target SNP position. Well, you use short read, and then you can determine, yeah, here's a SNP, here's a SNP, here's a SNP, and here's a SNP. You have 4 SNPs in your genes. And then you need to define the target you want to reconstruct. Okay? You have pairs of haplotype, then we use 1 or 0. They said, well, we really like the binary variable. You have 1 or 0 to represent the pair of haplotype. Okay? So this is a set of an order 1 or 0 here. Well, now we define what we want to reconstruct, and then we define what we have on hand. So you have data, you have reads. And then we introduced a read matrix here. And for each rows, we have a reads. And for each column, we have a SNP position. We have 4 SNP here, so we have 4 columns here. And you find that the green colors, the green rows, have more entries because you have smaller number of missing value. The long read cover more data. And in the rest of it, we have only short read. A lot of them are just missing values. So now we define Xij, the read matrix S with the numbers of read here, and we assign the value 2 or 3 if it is just an error, if it does not match to the SNP calling. Because remember, at here we know here only A or G. If it called T or C, we will give it C 2 or 3. So this is an error. And now the next thing we need to do is to define the similarity between the H and S. So you have data, and you make a guess of the H. Okay, you construct an candidate H, and then how likely they match together. We use an Xij here. If they match, you give 1 or 0. If they're an error, you give 2 or 3. And then this Xij will follow categorical distribution. So this is similar ideas we raised a couple years ago for the genome data. But here we have to introduce the transcriptome analysis as well. Then you have to consider the allele-specific expression. Well, to do that, we develop IDP-ASE. We want to reconstruct the most likely pair of the haplotype H and also the row. Okay? You want to estimate the row and reconstruct H. Well, unfortunately, row is continuous and unknown, so you cannot enumerate all the possibilities. Okay, even you want to do that, well, you have a lot of possibility. But we can draw the samples from the joint distribution of H and rows. Okay? Well, from the statistics 101, and you know this, this will proportional to the likelihood, and we just need to calculate this likelihood. And to calculate this likelihood, we assume each row are independent, each read are generated independently. Then you get products over each row. And then to account for the allele-specific expression, whether this read generated from the paternal allele or maternal allele, we have the mixture model, row and 1 minus row. It's generated from the paternal or maternal alleles. Okay? And then for each items, we can consider each variance are independent because they separate by many base pairs. So we consider this is independent. Then you can calculate their likelihood independently and the likelihood of categorical distribution. And with the parameter PJI 0, PJI 1, PJI 2, PJI 3. Okay?
So this independence is kind of supported by the notion of physical distance.
Mm.
How about the previous one? Based on the
the reads.
Technology.
Yeah, the
So
reads.
it makes sense to assume that they are.
Yeah, I think so. It really makes sense. Well, the reads generate independently.
Right.
Okay. Well, in the simple ways that we can calculate, we can estimate the Pij is the estimation of the error. Because given the base call, we have an error, how likely this is not true. So you can calculate from the sequencing quality score and then split it evenly to the other 3 nucleotides. However, we have a better model. Remember, we have a line QC give you an error pattern like this. This is, well, you can, if you check this carefully, you find that if you have an error rate, you cannot split it evenly to the other 3. So this is a finer error pattern you could have from the real data. This is generated by our tools Align QC. And then what remains is pretty simple. You can sample the joint distributions by MCMC, but here you recall that we have the sample rows, a continuous value, and also samples the binary haplotype, okay? Well, very typical MCMC here. And then we apply to a gold standard, the cell line is called the GM12878. This cell has been studied for many years. We know the haplotype already. So this is a gold standard haplotype we can use. Okay, come back to here. This is the accuracy of the haplotyping. And this is the number of SNP you have in your genes. Yeah, remember this is the random guess, right? So you won't make worse guess than this one if you have some data. Now, here is the result. We compare our result IDPSE to the other tools, M-base. Well, you develop a method, you definitely want to compare to the other existing method. Well, M-base is the dashed line, the solid line is IDPASE. Well, you definitely have a better performance when we use the hybrid sequencing data. Well, the red line is better than the blue line. But if you look very carefully, you have a dashed red line here, right? A red line in the red color. That means, well, if you use the M-base and the hybrid data, the same type of data, well, you cannot really get much better improvement here. Well, because in the M-base method, well, they use the similar idea of the read matrix, but they just use the marginals, the counts of the read in the matrix, cannot use the linkage information in the rows. So that's why if you use the M-base for the hybrid data, you do not have much gains in the accuracy, but ITPS is a better result.
So are you saying M-based basically treats long reads like short reads?
Yes. Yeah, because actually M-base were published about 2 years ago, and it only for short read, but you can put the long read into it, and then the output won't have much difference. Well, they just like increase the sequencing depth, increase the sample size, but they cannot use the linkage information in that rows. So that's why they do not improve the performance. And for the quantification, this is based on a simulation. Well, you do not have a gold standards of the quantification now, so we have to do the simulation. In the simulation, we find, well, when we use the red color, that is the long read only, TGS for third generation sequencing. We always have the highest error. This is the error, okay? The higher the worse, the lower the better. Well, we find that the green color always give you the highest error. Well, long read is not good for the quantitative analysis I mentioned at the beginning. And next, we find, the blue color, the third generation sequencing, short read can give you better estimation. Long read is good for quantification. Short read is good for quantification. And of course, the best one is the hybrid data, right? The hybrid data, the red color gives you the lowest error here. And for all 3 types of data, the solid line, IDPASE, is better than the dashed line, right? Okay. And here's what we simulate. We simulate a Gaussian-like distribution of row, and the estimation using different data size is matched to the simulation distribution. But M-base, they fails when the row close to 0.5, because they use a random guess of the phasing, right? So if you do a random guess and if the row close to 0.5, it's very difficult to distinguish 2 possibilities. Okay, we solved the quantification of the allele-specific expression, and also we determines the haplotypes. But we still have one problem to solve, the isoform level. The role here is the gene level. It's not the isoform. I never talk about any isoform level things in the model, right? But here it comes to the isoform level. Remember, this is the examples I present in the IDP. And originally, you only need to consider theta 1, theta 2, theta 3, because you only have 3 isoforms. But now, if you consider the paternal allele and the maternal allele, you have 6 isoforms. Now you have 6 parameters to estimate with the same data. But actually, you do not have the same data because if you use xi, you cannot distinguish the allele-specific expression. Now assume if you can estimate theta 1, 0, something like that, 6 parameter, you can calculate a value tau k. Tau k is the imbalance of one isoform between 2 alleles, okay, by this way. So how we calculate that? We use the same Poisson model, but we have to use some SNP information each short width. In this example, we have 4 SNP position, and for example, and then we label the regions by the isoform and also the haplotype. Okay, by this, because you have 2 isoform here, and then you have 2 haplotype, you use 4 index to label it. And let's take this as an example. Well, this R4W14 is show that, well, in this region 4, this is the 4 exon, and we have the A or C here. We have 3 V supporting C, so the value is 3 here, and then we have 1 V supporting A, so the value here is 1. The counting data we have is the allele-specific read number now. So instead of the X value, we have the Y value. Well, we also can assume this is a Poisson model. Well, of course, you can apply the negative binomial as the same. But, well, now you have to consider another parameter indicator here, whether it belongs to the reference haplotype or alternative haplotype. So this is the new variable you have to put into the model. But, well, again, this is a Poisson likelihood. You can find this convex optimization. So no problem. You can solve it. And once we solve it, we find something very interesting. Well, I'll show you some example. In this example of gene CORT, we have 2 SNP and 2 isoform. Each one express some exclusive X SNP. The first isoform express SNP2, the second isoform express the SNP1. So in this case, we just use a simple math that you can calculate the allele-specific expression. However, in this one, it becomes more complicated. They have some exclusive expression of the SNP2 and SNP3, but they also share the SNP1, right? Then you need a more complicated method to do that. And now, in number 3 example, we have a very complicated example. Well, you have SNP2 and 3 and also SNP1. You have 3 isoform, and 3 isoform express all SNP. SNP1 in the exon 1 region, SNP2 and SNP3 in the last exon region. They share the same. Actually, there's no hope to distinguish the allele-specific expression, right? But actually, we find SNP1 located in the flanking region of the junction. So if a V supporting this SNP1 and also an isoform-specific junction like this, okay, the red color, the green color, and the purple color, and this coverage can help you to distinguish them. And then we use these numbers of V's as the Y value in our model, in a Poisson model. Then we can calculate, well, we have the expression, the FPKM for the 3 isoform. We find the first isoform almost have no preference to the reference alleles or the alternative allele because it's close to 0.5. However, for the second one, is prefer more to the reference allele because it's 0.6. And the third isoform, you close to 0.3, it prefer to the alternative alleles. That means within a gene, you have 3 different patterns of the allele-specific expression. If you merge them together, calculate a gene level, you cannot see this. So this is very important to analyze transcriptomes by calculating the FPKM of each isoform and also their imbalance between 2 alleles. And when we apply to the breast cancer data and the human embryonic stem cell data, we find, for the human embryonic stem cell data, we have a distribution of this tau isoform level allele-specific expression at Gaussian-like distribution. If we set the threshold of 0.35 and 0.65, they call it significant allele-specific expression, we have some. But if you only calculate the gene level, you calculate rho, you find less because, as I mentioned, and the example, they will mask out each other. Okay, you will find less significant allele-specific expression. And we also find in the breast cancer, in the cancer data, because you may have the copy number variance, so you have more significant allele-specific expression. You see, this is much more significant and intense than the human embryonic stem cell. Okay, and here's a slide as a conclusion. What we talk about today is we talk about the identification and also the quantification of 2 dimension of a gene transcriptome analysis, gene isoform and haplotype. We need to calculate this value. We need to determine the isoform. We need to determine the haplotype. Okay, and we come back to this. These pipelines, we have LSC for error collection, IDP for isoform determination, and the allele specific expression. Actually, we have the other ones, but in each one, I just spend one slide to talk about that. APA, we have alternative poly-A. The alternative ending point of genes is kind of similar to the splicing event, and we develop IDP-ATA to determine this alternative ending point as well. Well, it's good timings. We're almost done. And well, this also contributes some complexities and also the different combinations of the genes. And for IDP fusion, we apply the long V and short V to determine the pairs of genes involved in the fusion event. We can determine the fusion site at a very high resolution in the single nucleotide resolution, and then we can predict very complicated fusion transcript or fusion isoform expressed in these fusion genes. And here's IDP de novo. Well, for non-model organisms, we develop a pipeline to assemble the transcript for the non-model organisms and annotate it and also quantify the transcriptome. While comparing to 7 other tools, IDP de novo have better precision, recalls, and F1 score. So this is some brief results. It's under reviews in bioinformatics now. And yeah, and at last, I really want to share this software to use because it's pretty useful. And when you input some long read data, and this is how the output look like. If we give you some HTML file showing you the mappability of your result, the length distributions, and the coverage annotation. This is the coverage, and also, well, like Elisa asked just now, well, how do you know the sensitivity of your data? So here's the fraction curve analysis showing that whether you generate enough data or not. And if not, if you still have a very significant improve, then you can generate more data to cover. And here's the error patterns we use for the error collection and also the allele specific expression as well. So I can tell you the homopolymer patterns of the error in the peptide data. And here it also gives you some number, how the error distributes in the insertion, deletion, and mismatch. And for all the figures, we have the PDF high resolution versions, so you can just download it and use it in your paper publication directly. So we almost there. Yeah, pretty good. Well, and now, yeah, you see, perfect timing. We have the acknowledgement slides. Well, I want to thank my collaborators in the peptide company. They generate a lot of data for us to run the proof of concept application. I want to thank my collaborator at Stanford, Vittorio Vanese, and his students, and Mike Schneider and his postdoc. They all provide a lot of help in generating data. And also, I want to thank the funding agents, NIH and Pharma Foundation for the young faculty. And also thanks my teams. They are very talented people from statistics, biology, and computer science. They help me a lot in developing these tools. Thank you so much for that. Yeah, I'm happy to take food and take questions.
So, we have one question.
Yeah.
I
And?
would take the one question.
Ben? Yeah, we can, and I can answer questions when you have food, right?
So, these novel transcripts that you found in where was it, in the stem cells?
Yeah, stem cell.
You had said that like some of them were 95% repetitive elements. By repetitive element, do you mean like high copy repeats? Like these are
A very good question.
Or do you mean like pseudogenes where it's a recent segment of duplication?
Well, we just check the UCSI checks, then you have a definition of multiply mappable, the mappabilities of regions. Once the mappability is smaller than 1, we call it repetitive.
I see, okay.
But you only compress strings of the same letter, right? You don't compress other type of repetition.
No. Yeah, because actually this is just a trick to the transformation of the sequence to help you to improve the alignment step. So actually do nothing in the error collection. Once you find this short way aligned to this long way, we decompress it and then to generate the consensus.
That's the thing.
Thank you.